Hello and welcome to Let's Crack NEET UG English. The first thing is we will be checking for the audio. Let me know if the audio is good. And as you can see on your screens, what we plan to do today is that is, we will be doing a recap of glycolysis, which we did in the previous lecture. Then we will talk about oxidative decarboxylation, which is the intermediate step, and then the Kreb cycle. Okay, so this is what we have planned for today. So let us wait for everyone to join. Till that time, all those who are there, just let me know whether this audio is clear or not. And then we will get started. Okay, Nitin, good afternoon. Is the audio clear? Hiren, good evening. Very good. Hiren says it is good. What about you, Nitin? Audio is clear. Very good. So students are just joining in, so let us wait for a few seconds more. And did you get a chance to go over the steps of glycolysis? Oh, okay, Priya, sorry, Priya, yes. So did you revise glycolysis, all the steps? Very good. Any doubt in that? Any doubt in that complete two-phase process? That is energy investment and then payoff, what happens where, and how many ATPs, NADH2s we are getting. Okay, no doubt, that's very good. So when we go over it, when we have a recap, you should be able to tell me the next step. Okay, Priya, no, everything is clear. That's very good. And how did you revise? Did you write that complete 10-step process, or did you just read it? Hi, Samuel. I'm doing good, Samuel. Thank you so much. Hiren says I made notes, and Priya says she wrote it down. That's very good. See, unless and until we write things, we won't be able to recall those things. So this is one important step when we study biology. And not only the cycles, but otherwise also, whichever chapter you study from biology, the best way is write down things and not like the whole paragraph, just the important terms or maybe just the steps. Reading NCERT is definitely important, but writing is the most important thing. And in writing also, it should be like once you read it, then close that notebook or book, and then maybe after half an hour or an hour, you should be writing. It should not be like seeing in the notes copy and then writing on a rough copy. It should not be copying. It should be learning, and then after some time, then trying to write it down. One chapter a day series is good, ma'am. Thank you. Okay, thanks, Samuel. You're liking it. I'm very happy. Okay, so this is like whenever we do a particular chapter, normally I keep telling my students about what should be done to learn a particular thing. When we study biology, it has to be done in a different manner. It cannot be done like any other subject. I have seen students reading NCERT, just reading NCERT, as if it is a novel, or you're reading an English book, or is it some kind of a social science or history book. Biology is basically the processes. So we need to draw, make the flow charts, then only the things become easier for us. Explanation is good. Thank you, Priya. Like when the students say that they're understanding, then it gives a satisfaction to the teacher also. Like whatever I'm doing is helping the students. So that is a great motivation for us also. Okay, now before we go over the glycolysis steps, the recap, tips on learning examples. Okay, that is Samuel's question. See, which example are we talking about? Samuel, can you give me a specific thing? Which example? Are you talking about morphology or any other chapter? Then I'll tell you how to remember those examples. It's hard, like morphology. Okay, I guessed it right. See, in morphology, there are so many modifications. When we come to that chapter of morphology, I will tell you tricks to remember the names, but I'm giving you one very general thing. In morphology, basically we are talking about root and its modification, stem and its modification, leaf and its modification, and so on. So if you're talking about seeds and fruits and roots and leaf and everything, try to find out, is it related with our everyday life? For example, if we talk of, say, wheat, we eat wheat every day. Rice, we eat rice every day. There are some vegetables like potatoes and tomatoes and chilies. These are the things which we eat every day. So you have to adapt to a technique that you talk to yourself in biological language, in the technical language. Say for example, I'll tell you one simple thing, that I made or I ate a vegetable, that is potato vegetable today, with chapati, and that potato was cooked in some oil and some mustard seeds were added into it, chili was added into it, and turmeric was also added into it. This is like general thing. Now if the same thing I have to tell myself, you don't have to tell it to anybody, you have to revise it on your own. So how are you going to talk about this whole food that you had today? You would say that I ate Solanum tuberosum, which was cooked in the oil of Brassica campestris by adding Brassica campestris seeds, capsicum and curcuma longa. You have used all technical scientific names, and this you eat every day. And every day the vegetable is going to be different. Today you are going to eat cauliflower, cabbage, brinjal, something. So if you do it, you don't have to mug those names up at the end of the session. Okay, families. In families also, suppose you're talking about Liliaceae family. In Liliaceae, when we talk of the economic importance, we are talking about so many things, onion and garlic and lilium and everything. So all around us, the same thing, you eat onion, you eat garlic. So use those words in your daily life. So for plant kingdom also there are important tips, but we'll do it as we come to that chapter, because right now I plan to give you some tricks about the Kreb cycle which we have planned to do today. So as and when we come to a particular chapter, I'll keep giving you the tricks or tips to remember certain things. So this is just the think how you should be studying biology. Not only it is going to become easier, but it will be interesting also. It is not going to become a burden for you. today I'm going to share some new slides with you because there are some new changes, new plans, and I guess you now know me, so no need to show this slide. The platform that we are using is India's largest learning platform, Unacademy, and I don't know whether you have installed this app or not, because on this app you get the special classes, the plus classes, the iconic classes, which I'm going to be talking about. Now this plus subscription, which is going to help you in getting daily live classes, unlimited access, structured courses and all those things. This is iconic. So there's one plus, one iconic. Here you get a personal coach. It is one-on-one guidance from the experts. There is going to be a plan for you, how and in how much time you will be finishing which particular thing, plus all the benefits of plus courses. So what exactly is in plus and iconic? The same thing: best educators, interactive class, structured courses, live tests and quizzes, plus the PDFs. Now in iconic you also get this benefit of personal coach, the planner, guidance and the analysis of your test, that where you went wrong. And this is the subscription plan. This is the fee structure. And you can see, if you go for a long-term plan, then there is a greater benefit, and because we all are planning for either NEET 2021 or 22, so according to that you need to take the subscription, whether you want to go for a 6-month plan, a 12-month or a 24-month. And this is the fee structure. And if you use this code of mine while you take the subscription, you will be eligible for 10% discount. This is for iconic. The previous one was for plus. Now in iconic, you know that you get a personalized tutor and there is analysis and all those benefits. So if you go for 1 year subscription, it is going to cost you 58,500, and if you use this code, then there will be your discount also. Benefits of plus are there in plus programs, and in iconic along with plus additional things. Alright, so this is the new thing which is coming up. They are starting with the iconic program where you know, along with everything else, now there will be a dedicated educator for you. So if you decide to study from a particular teacher, that particular teacher will be with you, helping you like on one-on-one basis. Alright, so Hiren has written I have. So I don't know what for are you writing this, so I guess it is for you have already taken the subscription. Hi Kalai. Okay, and we are going to have a recap of glycolysis. But before that, you've downloaded the app, but have you taken the subscription plan? If not, then decide on that fast. We started with aerobic respiration yesterday, and we said aerobic respiration has 5 steps. The first step was glycolysis. Then there is a link reaction which is called oxidative phosphorylation. The third step, that is Kreb cycle. Then there is electron transport chain, and the last step is oxidative decarboxylation. Decarboxylation. And the last is oxidative phosphorylation. Okay, and out of this, we have talked of glycolysis in detail. Now to sum up, what we studied in glycolysis was that it takes place In cytoplasm, how many phases? In how many phases is it completed? Energy investment phase and energy payoff. We are talking of phases. These are steps. What you say is right. There are 10 steps. 10 steps and 2 phases. Perfect. Now, do we remember what was the first step? First step was glucose undergoes phosphorylation. So from glucose we got glucose 6-phosphate. Here 1 ATP was invested. Glucose 6-phosphate changes into fructose 6-phosphate. Then fructose 6-phosphate changes into fructose 1,6 bi or diphosphate. And then this splits into 3-PGAL and dihydroxyacetone phosphate. And these two are isomers, that means they can interchange. The next molecule which can, or the molecule which can participate in further reactions is aldehyde. That is 3-PGAL. Now this 3-PGAL changes into 1,3-di-PGA. Now this is where oxidation reduction was taking place. Inorganic phosphate got added. Are you able to recall all these steps? So NAD gets reduced into NADH2, and this NADH2 is worth 3 ATPs. Then from this diphosphate, 1 phosphate is taken by ADP and we get 1 ATP, and here is 3-phosphoglyceric acid. Then the position of phosphate changes, it becomes 2-PGA. Then there is loss of water, it becomes phosphoenol pyruvate and then pyruvic acid, but before that 1 ADP takes 1 ATP. So 1 ATP here, 1 ATP here. So if you start with 1 PGAL, if you start with one molecule of PGAL, you get 1 NADH2 and 2 ATP. But because this is ultimately going to change into PGAL, so everything is going to be double. That means there would be 2 NADH2 and total 4 ATPs. 1 ATP will be used here also. So you will have to give those 2 ATPs back. So minus 2 ATP. So here we will get 2 NADH2 plus 2 ATP. This ATP is the net gain, the energy or the molecule which you can spend immediately. That is the net gain. And this is equivalent to 6 ATPs. So gross ATP gain is, gross ATP gain is 6 plus 2, that is 8 ATPs. What was the other name given to glycolysis, named after the scientist who actually gave us this pathway? What was the name? What is the name of glycolysis, named after the scientist? All those who said they revised? EMP, very good. EMP. What does E stand for? Kamlesh and Hiren, Kalai, they have given EMP. What is E? It is Emden. E-M-B-D-E-N, not R. Emden. Very good. Then M stands for Meyerhoff. M-E-Y-E-R-H-O-F-F. And P stands for the third scientist, that is Parnas. And that is why the pathway is called EMP. It takes place in cytoplasm. It is completed in 2 phases and 10 steps. 5 steps in first phase and 5 steps in another phase. And at the end, we will get 2 molecules of pyruvic acid. Okay? Parnas. P-A-R-N-A-S. Alright, now let us come to the link reaction. Because the next major step, again where we will be getting ATPs and NADH2s, is Kreb cycle. But pyruvic acid, cytoplasm of yeast cell, Kamlesh, what is your question? Cytoplasm of yeast cell. It takes place in cytoplasm of every cell. Whichever cell has cytoplasm, it would have enzymes for glycolysis, and that is where it is going to take place. Even in our case, this breakdown is taking place in the cytoplasm only. Alright. So now this pyruvic acid cannot participate in Kreb cycle. So there is a temporary and a short reaction which takes place, which converts pyruvic acid into acetaldehyde. This acetaldehyde can participate in Kreb cycle. So that formation of pyruvic acid, or rather conversion of pyruvic acid into acetaldehyde, pyruvic acid into acetaldehyde, or acetyl coenzyme A then, or let us write down the direct step, into acetyl coenzyme A. That is called oxidative decarboxylation. Now where is what happening? That we need to understand. Suppose this is a cell and we are drawing the nucleus in one corner because our main focus is mitochondria. And in mitochondria we know there are these folds, crista. Now in the cytoplasm, glycolysis takes place. So this glucose is broken down into 2 molecules of pyruvic acid. This pyruvic acid can cross the outer mitochondrial membrane. So if I draw only a part of mitochondria showing a crista, pyruvic acid can diffuse in this space. This is the outer membrane of mitochondria, and this is the inner membrane of mitochondria. The space between the 2 membranes is called outer compartment or perimitochondrial space. Perimitochondrial space. In this outer compartment, pyruvic acid is going to change into acetyl coenzyme A, and inner mitochondrial membrane allows the entry of acetyl coenzyme A, and this inner thing which is there is the matrix, and in the matrix would take place Kreb cycle. So this link reaction is basically of pyruvic acid getting changed into acetyl coenzyme A. So we can do this in one step, we can do it in two steps. We'll write both. Pyruvic acid, we have to remember we have done one glycolysis. That means one glucose has been broken down, and when one glucose is broken down, there are two molecules of pyruvic acid. This we have to keep in mind, because when we write down the energy gain, at many places we have to double everything. So one pyruvic acid changes into acetyl group. Acetyl group is a 2-carbon compound and pyruvic acid is a 3-carbon compound. That means one carbon must be lost. So here one carbon dioxide is given out. And now when one carbon dioxide is given out, that means now C3 compound has changed into a 2-carbon compound. But during this conversion, can we call it decarboxylation, loss of carbon dioxide? There is loss of carbon dioxide. But there is one more change which is taking place in this step, and that is redox. Something is getting oxidized and something is getting reduced. NAD is getting reduced into NADH2. What is NADH2? It is a check worth 3 ATPs. So one pyruvic acid will undergo the same process. The second pyruvic acid will undergo the same process. So should we write 2 here so that it is easier for us to calculate? So there are 2 pyruvic acid molecules because we are starting with one glucose. If a question is one pyruvic acid will give you what, then the calculation is going to be different. But because we have done glycolysis, glucose, 2 pyruvic acids, that's why we are taking 2. So we will get 2 acetyl groups, we will get 2 carbon dioxide, 2 NADs will be reduced to 2 NADH2, and when you say you get 2 NADH2, that is worth 6 ATPs. This number we have to keep in mind, because when we say aerobic breakdown of glucose gives us 38 ATPs or 36 ATPs. What have you heard? 36 or 38? Complete aerobic breakdown of glucose gives you 36 or 38. What is that number that you have heard of? It gives you 36 or 38. Hiren says 36. Anyone who wants to answer? "36" is the only answer that I have got so far. So at some places you find 36 written and at some places you find 38 written. So by the end of this aerobic respiration, we'll realize what is more appropriate. 36 is appropriate or 30? Oh, Hiren says 2 are used in shuttle system. Very good. So basically 38 are produced, but we are left with 36 because 2 get used up. And where are they used up? That we will see. Now these 2 acetyl groups which are formed, coenzyme A is added. And now we get 2 acetyl coenzyme A molecules, and they can participate in Kreb cycle. They can participate in Kreb cycle. So this is a link reaction between glycolysis and Kreb cycle. The location where it takes place is outer compartment. It is a link reaction between glycolysis and Kreb cycle. Why is this reaction required? Because pyruvic acid which is formed cannot directly take part in Kreb cycle. Why is it not possible? Because the outer mitochondrial membrane allows the entry of pyruvic acid, but the inner mitochondrial membrane is impermeable to pyruvic acid. So pyruvic acid cannot enter mitochondria, and all the enzymes needed for Kreb cycle, they are present inside the mitochondria, that is in the matrix. So pyruvic acid cannot go in, so it has to be converted into a substance which can enter inside the mitochondria and can participate in Kreb cycle. Alright, so now in this part, in the link reaction, is there anything which is not clear? Is there any difficulty in this part? Because the next step, Kreb cycle, is a big step, and at the same time it is complicated. So we will take 2 days to finish Kreb cycle. What we will do today is the introductory part, and I am going to draw the Kreb cycle for you in a simpler manner. And then once you are comfortable with the simple manner, then we will go a step higher. Oxidative decarboxylation. Okay, can anybody tell me the name of an enzyme which can be written here? If I have to write the name of an enzyme in this reaction, which enzyme can I write? You taught in very deeply. I learned so many new things. That's very good. Jainam says, no doubt, everything is clear. Perfect. I want to know the name of an enzyme which would help in this reaction. You know what is happening here. There are two things which are happening. So on the basis of what is happening, you can name the enzyme. See, I'm going to give you one name. From pyruvic acid, one carbon is lost in the form of carbon dioxide. So can we say that the enzyme is pyruvate decarboxylase? Because that carbon is coming out from pyruvate. So one enzyme can be written as pyruvate decarboxylase. Can we write one more enzyme where NAD is getting reduced? You can call the enzyme as NAD reductase. I want to give one more name to this enzyme here. This reaction, can I call it pyruvate dehydrogenase? NADH dehydrogenase. No, it is NAD is getting added with hydrogen. Dehydrogenation is removal. So NAD gets hydrogen and it is getting reduced to NADH. So it has to be NAD reductase, or hydrogen is getting added to NAD, but where is it coming from? It is coming from pyruvate. So pyruvate is losing hydrogen. So we call it pyruvate dehydrogenase. So if you understand the process, you can write down the names of the enzymes. Clear so far? This is the link reaction, oxidative decarboxylation. Now, let us come to Kreb cycle. Kreb cycle is also known as TCA cycle. It is also known as citric acid cycle. Now, why is it called Kreb cycle? This is named after the scientist. Why is it called citric acid cycle? Because the first compound is citric acid. Then what is this TCA cycle? Can anybody tell me what is TCA cycle? Why are we calling Kreb cycle as TCA? How many of you have studied Kreb cycle before? Jainam says tricarboxylic acid cycle, which is perfect and correct full form. But what exactly is meant by this? Tricarboxylic acid cycle. Tricarboxylic acid cycle. Now, what is this tricarboxylic acid? What do you mean by tricarboxylic acid? That is the full form of TCA, but I want to know what is meant by that. Is it some kind of an acid which is produced? Is it some intermediate compound? What exactly is this? Any tricarboxylic acid will have 3 carboxylic group. And what is carboxylic group? COOH. So, an acid which has 3 COOH will be called a tricarboxylic acid, and such a tricarboxylic acid is citric acid. So, you can call it citric acid cycle, not 3 carbon containing, 3 carboxylic group, 3 COOH containing the acid, and that 3 carboxylic group containing acid is citric acid. So you can call it Kreb cycle, you can call it citric acid cycle, you can call it TCA cycle. There are so many names which can be given. Now let us talk about the location. Location. Where does Kreb cycle take place? It takes place in the matrix of mitochondria. It takes place in the matrix of mitochondria. Now this cycle gets completed in 8 steps. So I'm going to just make a rough diagram or cycle for you so that you're able to understand how this is happening. Acetyl coenzyme A. From where did we get this acetyl coenzyme A? 1 glucose gave us 2 molecules of pyruvic acid, and 2 pyruvic acids got converted into 2 acetyl coenzyme A's. We are talking about 1 acetyl coenzyme A, and then we will double it. Acetyl coenzyme A, we have to remember it's a 2 carbon compound, and it is accepted by a 4 carbon compound. So you will get here a 6 carbon compound, and that is citric acid. this citric acid will change into its isomer, and that is called isocitrate. Very simple. Citric acid is 6 carbon, isocitrate should also be 6 carbon, they are isomers. Then there is loss of carbon dioxide. One carbon dioxide is given out. That is decarboxylation is taking place. So if one carbon is lost from a 6 carbon compound, we get alpha ketoglutarate. Alpha ketoglutarate is a 2 carbon compound, is a 5 carbon compound. One carbon is lost from the 6 carbon. Then it changes into isocitrate, which is a 4 carbon compound. That means one carbon is lost. It changes into citrate, which is again a 4 carbon compound. Wait, it is an okay, so this is a 4 carbon compound and this is also 4 carbon compound. Then this changes into fumaric acid, then malic acid, and then this 4 carbon compound is regenerated, that is oxaloacetic acid. Alright, so this is 4 carbon, this is 4 carbon, this is 4 carbon, and oxaloacetic acid is also a 4 carbon compound. Succinic acid or succinate. So at 2 places carbon dioxide is released. Now I am going to number these steps. This is step number 1, 2, 3, 4, 5, 6, 7 and 8. So it is completed in 8 steps. Now we have just written the compounds: citric acid, isocitrate, alpha keto, succinate, succinate, isosuccinate, fumarate, malate and oxaloacetate or oxaloacetic acid. This is from 1 acetyl coenzyme A. Now reaction numbers or step numbers 3, 4 and 8 are redox reactions. Which are we talking about? 3, 4 and 8. Redox. That means something is going to get reduced and something is going to get oxidized. What is getting reduced is NAD. So if NAD gets reduced, it will be reduced into NADH2. Now can we add this here? NAD getting reduced into NADH2. Step number 3. At step number 4, NAD gets reduced into NADH2. Step number 8, NAD gets reduced into NADH2. 3 steps where redox is taking place, and those steps are 3, 4 and 8. 3, 4 and 8 are redox, but you have to remember which is that substance that is getting reduced. Step number 6 or reaction number 6 is also redox, but here FAD gets reduced into FADH2. Which is step number 6? That is this one. So FAD is getting reduced into FADH2. Okay, what we have to remember is NADH2 is worth 3 ATPs and FADH2 is worth 2 ATPs. So basically we are getting some molecules which will later on give us ATP. One more reaction or step number 5 gives us 1-phosphate. Actually, ultimately we will get ATP, but that is not directly. In step number 5, what happens is there is a substance called GDP. ATP is adenosine triphosphate. This is guanosine diphosphate. It takes the phosphate, changes into GTP. And when this GTP breaks into GDP, it releases the energy which is taken by ADP and we would get ATP synthesized. So in one Kreb cycle, what did we get? We got 1, 2, 3 NADH2. We got 1 FADH2 and we got 1 ATP. Okay, 1 NADH2, second NADH2, third NADH2 and 1 FADH2. Let us calculate the ATP. 3 NADH2 into 3. How many ATPs? 9. 1 FADH2 into 2. One molecule is going to give us 2. That is 2. And 1 is ATP. So this is anyways ATP. How many ATPs do we get from one Kreb cycle? That is 12. And we have 2 acetyl coenzyme A's. That means they will go 2 Kreb cycles. There will be 2 Kreb cycles and these 2 Kreb cycles will give us 24 ATPs. This is when you are talking from one glucose molecule. Now, have you drawn this cycle? If yes, then tell me. Here there is a coenzyme also which gets added. So it is succinyl coenzyme A. Okay. And then succinate. Alright. So did you write this complete cycle? And do you think you will be able to remember these names? Okay, Hiren says yes. So how many of you think you will be able to remember these names? Succinic acid, isocitrate, alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, malate, oxaloacetic acid. Okay, so I got the answer only from Hiren. What about others? And this is the important thing which we have to remember whenever we talk of Kreb cycle. Mnemonic would be handy, okay? All right. Can you remember this just one thing? Acetyl coenzyme A. Can we remember this first step? Then I'm going to tell you a trick to remember this. Say I'm a teacher. So I'm going to tell you, see, I know some simple formula making options. So I just said a sentence. Did you hear it carefully? I said, see, I know some simple formula making options. Will you be able to remember this sentence? Kalai says, try to remember by trick. What is your trick? How do you remember? Oh, Hiren seems to like this sentence. So as a teacher, I can tell you, see, I know some simple formula making options. And now, can we write down the names? So this is citric acid. This is isocitrate. This is alpha ketoglutarate. This is succinyl coenzyme A. This is succinic acid or succinate. This is fumaric acid or fumarate. This is malic acid or malate. This is oxaloacetic acid or oxaloacetate. And then the next step is number the steps. See, I know some simple formula making options. This is what we have to remember, and then we have to remember the numbers. 1, 2, 3, 4, 5, 6, 7 and 8. And I have told you that step number 3, 4, 8 are redox, where NAD is getting reduced into NADH2. This is point number 1. Step number 6 is also redox, but here FAD gets reduced into FADH2, and third thing is step number 5 is going to give you 1 ATP, but not directly. So if you remember this, you will be able to answer all the questions which are based on Kreb cycle. Now let me tell you what kind of a question can be asked on Kreb cycle. Your way of writing the cycle is much better. See, when we if you are in 11th class, then you will have to draw this entire cycle and this is going to be asked as a 5-mark question. And if you miss on something, then it is totally going to be messed up. So for your 11th class exam also this is good, and for competitive, which I am telling you right now, it is going to help you answer the questions in very, very less time. See, when we are targeting competitive exam, we don't have to write this. We have to simply write, see, I know some simple formula making options. Okay, this is what we have to write. Then 1, 2, 3, 4, 5, 6, 7 and 8. So if in a competitive exam the question asked is that you have one molecule of alpha ketoglutarate, how many ATPs will you get? Tell me. You have the Kreb cycle in front of you. Tell me how many ATPs will you get? And in how much time can you answer this? If you are given one molecule of alpha ketoglutarate, how many ATPs will you get? Everything is right there in front of you. What is going to be your answer? Are you still calculating? You have one molecule of alpha ketoglutarate. How many ATPs will you get in the Kreb cycle? Hiren has written 12. Do others think that this answer is correct? See, in step number 3, 4 and 8, you would get 3 ATP, 3 ATP, 3 ATP. In step number 6, you will get 2 ATP, and in step number 5, you will get 1 ATP. So, you are starting from here. So, how many ATPs will you get? You won't get 12, right? So, this step is not there. You are starting with alpha keto. So, 3 and 1, 4, 5, 6, 7, 8 and 9. This is how you're going to answer. And how much time is it going to take? Not more than 20 to 30 seconds. Okay. Now, listen to the question. Next question. Second, if you have one molecule of succinyl coenzyme A, how many ATPs will you get? One succinyl coenzyme A. How many ATPs will we get? Hiren says 6. What about others? Do they also think 6 is correct? Because you are starting from here. So 1, 2, 3, 4, 5 and 6. Okay, you have one fumaric acid molecule, one fumaric acid. How many ATPs will you get? Kalai has also said 6, okay. If you have one fumaric acid molecule, how many? Larry is also saying 6. For the previous question, if you start with fumaric acid, how many? Just 3. Because you are starting from here and this is 1. You have one oxaloacetic acid. Answer. Fourth, you have one molecule of oxaloacetic acid. How many ATPs will you get? Samuel, your answer 3 is for the previous question, right? If you have one molecule of oxaloacetic acid, then, okay. So how many ATPs will you get in the Kreb cycle? Larry says 1. I think Hiren also says 1. Check. If you have one molecule of oxaloacetic acid, then how many ATPs will you get? If there is one molecule of oxaloacetic acid, don't you think it will bind with acetyl coenzyme A and the complete cycle will take place? So the number of ATPs are going to be 12. Clear or not? If you have one molecule of oxaloacetic acid, that is the place from where the reaction starts. And if you have one oxaloacetic acid, it will bind with acetyl coenzyme A and the complete Kreb cycle will take place. So one complete Krebs cycle gives us 12 ATPs. Is it clear how we got 12 ATPs from one oxaloacetic acid? Ashutosh is asking what is glycogen. It is the reserved carbohydrate in animals. Anyone who has confusion in 12 ATPs? He didn't understood. What about others? If you have one oxaloacetic acid, then this whole cycle is going to take place. And if the complete cycle takes place, the number of ATPs that we get are full, that is 12. It is understood that if you have the starting point, then obviously the whole cycle will take place. Okay, acetyl coenzyme need not be mentioned here. Clear? Okay, so now this is a Krebs cycle and I have given you like 50% of the Krebs cycle part. But the most important thing is now you can remember the steps, the compounds which are formed by the sentence which I gave. And I hope you have written down that sentence. Anybody who missed out on that sentence, I can tell you that sentence again in the next class. And now when we meet tomorrow, we will draw the Krebs cycle and we will try to add the enzymes in it, so that the complete cycle is done, and then we will also know which enzyme is responsible for which reaction in the Krebs cycle. Any doubt, any question on Krebs cycle? Go over it, draw it one more time, write down the steps and remember in which step what happens. And when we meet tomorrow, we will be able to discuss this cycle again. I guess no doubts, because it is too early to ask for doubts. Our doubts are going to be there once you study this cycle at home. आशुतोष ने पहले पूछा था what is glycogen? Now what is glyphosate? What exactly are you trying to ask, Ashutosh? Glycogen I have already explained. In our body, the carbohydrate, that is glucose, is converted into glycogen and is stored in muscles and liver. Okay, so this is what I had planned for today's class. So in the next class, that is again tomorrow same time, 3:30 to 4:30, we will start with Krebs cycle, add few things into it, solve similar type of questions, and then continue with the next step. So don't forget to revise the cycle, don't forget to draw it, and try to remember that sentence which I gave you. Alright, bye for now. I'll see you tomorrow.